Hello, everyone. Before I get started on my lecture today, I just wanted to let you know that I will have Zoom office hours from now on Monday, Wednesday, and Thursday from 1:30 to 2:30. If you go on to Canvas, there is a module that says Johnson office hours. There is a link, and you press on that link and it will take you to my office hours, okay? Make sure that you only go on these days between these times, otherwise you won't see me there. If these times don't work for you, please email me and we can set up an appointment, okay? And we can talk face-to-face over Zoom so that I can kind of help you any way that I can, all right? If you have any questions about Zoom, just let me know and email me. All right, so we are now going to start our next lecture, and this next part is going to be about patterns of inheritance. So this is going to go over how we kind of can tell things about these traits that we inherit from our parents and from our grandparents, how that actually works, and some specific examples about different types of inheritance that we know about. There are some features that kind of make you the person that you are that are solely determined by genes, okay? And so we'll say solely determined by genes, okay? And so remember, genes are going to be these segments of DNA that code for proteins and other things like that, okay? There are going to be some other types of features that are going to be determined by the environment. Okay, so for example, if you look at my fingernails, if you look at this, it looks like I have, dark maroon fingernails. That is determined by the environment. I was not born with this color fingernail. I painted my fingernails this color and I can remove it. And so that is an environmental feature that I can kind of play around with. Your hair color is also something like that. You can get tattoos. Those things are going to be determined by the environment. There's something that you are actually doing, and they are not things that we would consider inherited because, if I have a daughter, she's not going to be born with this color nail, even if I was wearing this while I was having the baby, because it's not something that's inherited. So we're going to talk about things that are inherited today. And one thing I want you guys to think about, kind of this connection between biology and your lives, so we'll say society, is most people love dogs or cats. And we know a lot about dogs and cats and their genes and their different traits that they can have. Okay, people pay thousands of dollars to pay for or to buy specific breeds of dog and cat. And humans have selected for different types of breeds over the last, we think about 15,000 years. Okay, so we've been breeding dogs for probably about 15,000 years from what we can see in the historical record. And basically all we're doing is genetic tinkering. And what that means is we are selecting for traits that we want. So do we want a dog that is nice and is not super ferocious and is going to bite us at all times? But do we also want a dog that will be kind of aggressive towards individuals it doesn't know? So all of these things we're selecting for these behaviors and appearances that we like. And this is what we're going to talk about today, about this inheritance. So how does that actually work? So the big main topics we're going to talk about today are genetics and hereditary. Okay, and so we need to know what genetics and heredity mean. So heredity is the transmission of traits from one generation to the next. So when we say things like inheritance, you inherit your mom's eye color or her hair color. You inherit these different traits from your parents, okay? The field of genetics is going to be kind of the scientific study of heredity. So this is going to be, is the scientific field scientific study of heredity. Okay? And so what we're going to talk about first is who we will call the father of genetics. And the father of genetics is going to be this man right here. This is obviously a cartoon, but this is Gregor Mendel. Gregor Mendel is going to be a monk. He's Austrian, and he was a monk, so he lived on a monastery. And he is going to kind of be this first figure that really starts to understand genetics. And he's going to do this at a time where we don't know about chromosomes, we don't know about genes and things like that. We don't know about evolution, but he's going to use this pea plant that you see pictured here to kind of help us understand how genetics works. Okay? So we'll kind of hear a little bit about Gregor Mendel. Okay, so he's going to work in the 1860s, so quite a long time ago, and he basically is going to look at how parents pass on their offspring discrete genes. Okay, he's not going to use the word genes, but he'll say things like heritable factors. Okay, so he basically is trying to understand, if we look at the parents, and this could be the parents of a plant, so the two kind of plants that come together to fertilize and then create a new plant, how are they passing on their different traits to the offspring? Okay, and we now know that genes are going to be responsible for this. Genes responsible for inherited traits. So like the reason why your eyes are the color that they are is going to be a combination of what you get from your mom and your dad. Okay, and so those are going to be genes that we'll start talking about. The inherited traits can be things like skin color, height, eye color, things like that, that we'll talk about. Okay, genes retain their individual identities, generation after generation. So if you've ever been told that you look like a relative other than your parents, someone like, oh, you look just like your grandfather or your grandmother, what's happening is that there are genes that are passed on, and those are being passed on to you. So you are kind of the culmination of both your parents, but then also their parents, and then the parents of their parents. So those genes are going to be passed on from generation to generation. And that's why you can kind of track certain traits in families. Okay. So Mendel is going to work on a group of, he actually works on a couple different types of plants, but most of his experiments that he found most of the information about the genetics is going to be in the pea plant. So we're going to talk about Mendel and his pea plants. Okay, and so we were talking about peas. This is a pea plant right here. They're flowers. They're going to have, like the kind of peas that we will eat. these are the plants he's going to use. Okay, the reason why he uses peas is that they are easy to grow and they come in multiple different varieties. They come in many readily distinguishable varieties. So I'll talk a little bit about what I mean by varieties, okay? So one of the things that we're going to start talking about are these things called characters, okay? So I'm going to define what a character is for you. And these are the kind of the vocabulary that Mendel used in his work. And a character is going to be a heritable trait or feature that varies among individuals. Okay, so I keep on giving the same examples, but eye color, it varies from person to person. How tall you are varies from person to person. These things are what we would call characters, okay? And then within a character, you have what we call a trait, okay? And a trait is going to be a variant of a character. For example, so if we're talking about eye color, so that's our character, we could say that there's different traits. So you could have brown eyes or blue eyes. We know it's more complicated than that, but for now, this is something we can just kind of make simple. Okay, so the eye color would be the character, and then the trait would be, well, you have brown eyes or you have blue eyes, okay? Now, one of the reasons why Mendel chose pea plants is that you can readily kind of control the actual pea reproduction. Okay, and so what happens is, here's a pea plant flower, and we haven't talked really about flowers or plant reproduction at all, but just a little kind of overview of that. So for most flowers, there is a female part and male parts. So the female part is the center part called the carpel. It produces eggs, and there are eggs down in this part, okay? Then we have these other little things. These are called stamens. Those are gonna be sperm producing pollen. And so what Mendel was able to do is he could control like what fertilization was taking place. So he could take one flower and cut off all the stamens, but leave the carpel with the eggs, and then bring in the right type of pollen, which is sperm, to the plant that he wanted to look at. Okay, and so this allowed him to really control what was happening. So I'm going to show you what this looks like. So what he would do is he would remove the stamens from a purple flower, and we're going to start talking about kind of purple versus white flowers. So flower color will be our character, and then our traits could be purple or white. So he would take a purple flower, he cut off all of the stamens, and then he would take a stamen or the pollen from a white flower and put it on the purple flower. Okay, and when he would do that, he would then produce, so there would be fertilization, and a pea pod would be produced. Those peas can then be planted, and we can then look at what are the flower colors of that next generation. Okay, and so we're going to be talking about this kind of experiment multiple times in this lecture. Okay, but this is one of the big reasons why he used pea plants, is because they could easily be manipulated in this way. You cannot do this with humans. You cannot manipulate who mates with who or those kind of things. So, or many animals, it's more difficult to really control for that. So plants make a really good resource for this. Okay, so a little more detail about the plants. So what Mendel did is he created what we're going to call purebreds. Okay, and I've recently been watching Harry Potter over again. Purebreds are like what you'd find in Harry Potter, but it mainly means that purebreds are going to be plants that will only produce the type of seed that looks identical to them. So I'll write this out because it's a little bit kind of confusing. So purebreds, okay, so they are plants that will produce only seeds that produce plants that will look exactly like the parent plant. Okay, and so what we mean by this is that if we have a purple flower and we do crosses between purple flowers, every time that you make the seeds, they will always produce purple flowered plants, okay? So pure breed means that a purple plant will only make purple plants. And we could have white, so white plants would only make white plants, okay? This is gonna be really important because you have to have these pure breeding groups so that you can then start to see what happens when you mix them, okay? So this is kind of an example of that where we have pure breeding purple, pure breeding white, and what happens when we look at when we cross this. And you're gonna see that they're all purple, and we're gonna explain why that happens, okay? So what Mendel would then do is he would do these things called kind of crossbreeding experiments, okay? So Mendel would cross, which basically means mate, purebreds, and see what the offspring looked like. Okay? And when he would do this, what the offspring would be considered, they would be considered hybrids. Okay? So hybrids are going to be the offspring of two different purebred varieties. Okay, so if we have purple, oops, that's not spelled purple. Purple flowers would be one pure breeding group, and then we would cross it, and the way we write across is just an X with white flowers. And see what happens when we do that. Okay? When we do this, so the cross fertilization, itself is referred to as a genetic cross. Okay, so you're gonna hear this word cross a lot. So what happens if you cross white flowers with purple flowers? Okay, when we start doing these crosses, you're gonna start to see kind of a very specific way of how we represent the different groups. Okay, and so how we're gonna do this is that we will discuss the parental plants, and we are going to call them the P generation. Okay, we will then call the hybrid offspring the F1 generation. Okay, and this F stands for filio, which means son or daughter. So it basically means the offspring, okay? We can then do another cross, and I'm gonna write out all of this so you can see what this would look like. A cross between two hybrids, or the F1 generation, would lead to what we call an F2 generation. Oops, sorry. And so what Mendel would do is he would track kind of the different generations of these crosses. Okay, so Mendel tracked inheritance of characters such as flower color. Okay, and he was able to come up with a variety of hypotheses about kind of based on this. Okay, so first I want to just to kind of show you what, for the specific pea plant that he looked at, these are the different characters that he was able to look at. So the pea plants had two different types of flowers. So it could either be purple or white, okay? There was different flower position, so depending on if it was at the end, which we call terminal flower position, or kind of off to the side, which is axial. The seed colors were either yellow or green. The seed shape was either round or wrinkled. And then the pod shape, pod color, and stem length all were gonna have similar situations where there's two options. So you'd either see purple or you see white, you see tall or you see short. Okay, and so we're gonna start talking about, we're mainly gonna talk about flower color and these two things down here, seed color and seed shape, and specific examples, but you could see this in all of these examples. Right now, I'm not going to discuss what dominant and recessive means. We will come back to that, but we will learn kind of what that all means. So what I wanted to kind of show you is I'm gonna first show you in a picture and then I'll kind of show how this happens in an actual cross. This is how we would write out this is a hybrid cross. Okay, so we're gonna take purebred parents. So here's purple purebred flowers, white flowers that are purebred. When we cross those two, so we can pollinate, a purple flower with some white pollen, we then get this F1 generation. And every single plant that comes out of that F1 generation have purple flowers, okay? So that's gonna start to be important. So why are they all purple? Why do we not see any white flowers? When we then take these purple flowers from this F1 generation, okay, these are hybrids, we then cross them with each other. So we could take one flower, from this generation with another one, cross-pollinate, and then what we see is that in the next generation, what we have is we have some purple flowers, most of them are purple, but then we also will have a white flower show up. And the ratio is always the same. So there's usually, if we kind of look at the numbers, 75% of the flowers will be purple and 25% will be white. Okay, and so Mendel did this experiment with all of these different kind of characters and was starting to see that these ratios kept on coming up over and over and over again. So what's actually happening is that we are seeing kind of what we're going to start calling Mendel's laws of kind of inheritance. Okay. So I'm going to first kind of talk about kind of what these laws are, and then I will actually start to do some crosses so we can see what these crosses would look like. Okay, so let's say Mendel's, oops, and of course I spelled his name wrong. Sorry, my spelling has not gotten better over this time in isolation. Okay, Mendel's law of segregation. Okay, so when Mendel did a cross, so when we set this up, so Mendel does a cross between a purple purebred, like we talked about, and a white. What he found is that that first generation, like we saw right here, all of them ended up being purple, okay? And he found that the white flower, the gene for the white flowers doesn't disappear. So it's still there because it will all of a sudden show up again in the F2 generation. So Mendel found that the gene for white flowers did not disappear in the F1 plants, but was somehow hidden or masked with the purple flower factor was present. So something about this purple is overshadowing this white. And so when they come together, you're not being able to see it. Okay. And so from that, he kind of came up with this idea that these F1 plants must have carried two factors for flower color character, okay? And there's one for white and there's one for purple, okay? And so when they're together, you see just purple, but then you can kind of get just white flowers and purple flowers in this F2 generation. So based on this result, what he saw, he came up with 4 hypotheses that are going to kind of start to create these laws of segregation. Okay, so the first hypothesis that he came up with is there are alternative versions of genes, and we're going to start using genes because this is what we now know. He wouldn't have used the word genes, but this is the technical term, that account for variations in inherited characters. Okay? And so he would have called these different versions, you know, these traits, but these alternative versions are going to be called alleles. And this is going to be an important term, okay? So the alternative versions of genes are called alleles. Very important word, okay? So these alleles, when we're talking about, for example, this experiment that he did, the alleles would be, well, there's a purple allele and there's a white allele, okay? So they're the different versions of what you can see. Okay, so that is going to be kind of one of his first hypotheses that he comes up with. His second one is, for each inherited character, an organism inherits 2 alleles, one from each parent. Okay, and so hopefully this is starting to kind of remind you of what I was discussing when I talked about when I talked about meiosis, because we would talk about how you would have, you know, you have your mom's chromosome and your dad's chromosome, and we call these homologous chromosomes. And so you are getting 2 of each, okay? So when we're talking about specific genes, you'd have one that you get from your mom and one you get from your dad, and those would be then alleles, okay? So it's the version of the gene that you got from your parent, okay? Now, depending on the situation, you can either have identical alleles. So for example, you could have 2 alleles that code for purple flowers or 2 alleles that code for white flowers, or you could get one of each, so a purple and a white, okay? And so we have names for this, okay? So an organism that has 2 identical alleles for a gene is said to be homozygous for that gene. Okay, this is a very important word, homozygous. Homo means same, okay? And zygous is referring to the zygote. And so what this is saying is that these individuals will have identical alleles. So for the purple ones, it would be 2 purple alleles. If you have the white version, it'd be 2 white alleles, okay? Now, this other example that I talked about, when they have one of each allele, that's gonna have a different name, okay? And so when an organism that has 2 different alleles for a gene is said to be heterozygous for that gene. Okay, heterozygous is also a very important word. Okay, the word hetero means different. So that means that there are 2 different alleles. So that is going to be kind of another important term. So allele, very important word. Homozygous, very important. Heterozygous, very important. And homozygous and heterozygous are basically saying what kinds of alleles do you have. Do you have 2 of the same, or do you have 1 of each? All right, for his next hypothesis, he basically starts to talk about what happens when you have 1 of each. What allele will be what you see in the individual. Okay, so if an organism has 2 different alleles, which means it's heterozygote for a gene, 1 allele determines the organism's appearance, and it is called the dominant allele. Okay? So what this is saying is, and we saw this already in this example right here, if you have a purple and a white and you cross them, the next generation is all purple, which means that the purple allele is dominant or overshadowing the white allele. Okay, so purple is dominant to white. So we call that the dominant allele, but what do we call the allele that's being overshadowed? Okay, so the other allele has no noticeable effect on the organism's appearance and is called the recessive allele. Right? And so in the case that we talked about, the white one would be the recessive. Okay, so this would be recessive and this would be dominant. The only way you can tell which one is which is when you do a crossing of the purebreds. Okay, because then the purple flowers and the white flowers, if they have a cross and all of them turn out to be purple, then you have a situation where purple is dominant to recessive. So I want to bring back this picture that I showed you earlier of these different traits. And so what this dominant and recessive thing means is that purple is dominant to white. Axial flowers are dominant to terminal. Yellow seeds are dominant to green seeds. Round seeds are dominant to wrinkled, and so on and so forth. And so in his experiments, he discovered which one was the dominant and which one was recessive by doing those simple cross breeding experiments. All right. So when we start to kind of go forward with talking about these different crosses, we're going to start using letters to kind of represent the different allele types. And so the way that geneticists do this, okay, so geneticists, this is just kind of how they do it. Okay, so they use uppercase letters for dominant alleles. And I'll show you what this looks like. Oops. There's too many L's there. And lowercase for recessive. Oops, cis. Okay, so for example, and you always use the same letter. So if we're talking about, for example, round versus wrinkled, okay, for this one, we would choose some letters, and you want to choose letters that are easy to tell the difference between an uppercase and a lowercase. So for round, we would give the round allele a capital R, and our wrinkled one, we would give a little r. You do not use two different letters. You have to use the same letter, but just uppercase versus lowercase. Okay, so we can make up, you know, I'm going to have examples where we're going to do purple versus white, and you can use capital P for the purple, lowercase p for the white, and so on and so forth. So you can but you always use the same letter, okay? And this is just the standard kind of way that geneticists do it. Okay. We have our fourth hypothesis, and then I'm going to actually start to show you how to do a cross. Okay, and so this last one says a sperm or egg carries only one allele for each inherited character because the two alleles for a character segregate or separate from each other during gamete production, aka meiosis. So again, remember when we talked about meiosis, we talked about how we end up with half of the chromosomes in a sperm and an egg. Okay, because when they come back together, then they're going to make a whole organism, but you have to have a reduction, so they're only going to carry one allele. Okay, and so this is the actual law of segregation. Okay, this statement right here. So this is the law of segregation. Okay, when a sperm and egg unite at fertilization, each contributes its alleles, restoring the paired condition in the offspring. So what that is just saying is that once you have fertilization take place, you then will have two copies. You'll have one that you got from your mom and one you got from your dad. Okay, so I wanted to show you what the actual cross between kind of this purple and white plant example, how we would actually write this in kind of a way a geneticist would do, okay? So we would have a purple flower plant, and we would have a white, okay? And I want to remind you that these are pure breeders. That means that when you breed the white pure breeders together, they will only ever produce white plants. And when you breed purple flower purebreds together, they will only produce purple, okay? They have to be pure breeding, otherwise we start to get these weird ratios. So how we're going to actually start to kind of talk about this is I'm going to give the capital letter P as our dominant letter for this allele. Okay, so purple flowers have to have the dominant letter. And because they are going to be pure breeders, that means that they're going to have two capital P's. Okay, so they're going to be homozygous dominant because they're going to have the dominant traits. Okay, the white flower is going to have little p's. Okay, so they're going to be a homozygous recessive, so they have the little p's. Okay, so big P equals purple, little p equals white. Okay, and so we can have different types of allele. We always have two of each, so we can have a big P, big P, little p, and little p. Okay, and we know that this one is white, this is going to be purple, and this one's also going to be purple. Okay, so this is just what we know from these experiments being done before. Okay, so this is what the cross looks like. And so I'm not right now going to tell you who's the mother or the father. It doesn't really matter in this case. We will sometimes talk about that because it will matter. But the first thing we have to do is we have to figure out what kind of gametes could these make, okay? And so basically when they only have only one letter, so they have capital P's, that's the only type of sperm or egg they can make. So I'm going to say this is a gamete. Okay, so when they were going through kind of meiosis, let's just say that here's a chromosome that has the allele for purple color. And so they would have a big P and a big P on their two homologous chromosomes. And one would go into one sperm or egg, one would go into other one. And so these would be then in the gametes, okay? For white flowers, they would have little p's, okay? Because you'd only be able to do little p's. And I'm gonna draw a line under that to tell, sometimes it's hard to tell what a little p versus a big P is, okay? So what we're gonna now do is something called a Punnett square, which is basically showing you the probability of what the offspring could be. And when you do this, you're gonna use kind of the gametes that could be produced by this one plant. And so they're gonna be able to make big P's. And there's actually two of them because we know that there's two alleles, so they're gonna be segregated. So we have two P's right there. And the white flowers are going to be able to make these lowercase p gametes. So I'm gonna write them together on this side. Okay, we then have in each of these boxes is going to represent what happens if a sperm and an egg came together and fertilized. Okay, and so they would get a big P from either the sperm or egg and a little p from the opposite. Okay, and so you would just kind of basically write what the combination would be. So it would be big P, little p. And just a little bit of how this is standard, you always write the capital letter first and the lowercase letter second, okay? I could have put these two P's here and switched these, it would be identical, okay? So what happens is in this first cross, and so this is the P1, this is the parental generation, okay? This is gonna be our filial one, our F1 generation. All of the offspring are going to be big P, little p, okay? And because purple is dominant to white, it's gonna overshadow it. All of them are gonna be purple flowers. Okay, now our next cross could be like, well, what happens if we take two of these individuals? So we take one of the plants, right here, and we can take this plant right here, and we do this cross. Well, we always have to know what are our gametes gonna be, and so in this case, they can make gametes that are big P's and gametes that are little p's. This one could also make a gamete that's a big P, and this one can make a gamete that's a little p. Okay, we can do another cross. Okay, we can put big P, little p, so I'm putting those ones right there, big P, little p, and then I can do my cross. So big P, big P, big P, little p, big P, little p, and little p, little p. So then I can look at what I have. I have one big P, I have two big P, little p, and I have one little p. And then I could say, okay, what does this look like? So big P is gonna be purple. Big P, little p is gonna be purple. And little p, I should write, oops. to do that, is going to be white. Okay, and so what I could do is I can actually come up with a ratio of purple to white. So there's gonna be 3 purple to 1 white flower. Okay, so if we did this, we're obviously not gonna just have 4 plants. We would have, hundreds of plants, and then we could count how many of them are purple, how many of them are white, but we would always end up with this ratio right here, which is 3 to 1. So a little bit about what I just did. So this is called a Punnett square, named after an individual that kind of created these. And we will be doing a lot of these in lab. So you'll get good practice, but let me just tell you a little bit about Punnett squares. Just make sure I spell it right. Okay, so they're a tool that helps us kind of look at the 4 possible combinations of gametes. Okay, and then they will show you what are the 4 resulting offspring. And I should say possible offspring. Okay, because for example, every time that you have a child, you have a 50-50 chance that it's a girl or a boy. Okay, and so it's not that every single time, you've had one girl, so now it's your turn to have a boy. Every time there's going to be some type of ratio, and it's always going to be 50-50, to have a boy or a girl. Okay, so each square represents an equally probable product of fertilization. Okay. So when we start to talk about these different kind of what things look like and then looking at their alleles, we need to come up with 2 different terms to discuss them. Okay, and so we need to be able to distinguish between kind of what an organism looks like and what its genetic makeup is, okay? And so we're gonna start using these important terms. So the first one is gonna be phenotype. Okay, so really important term. This is gonna be the physical appearance. Okay, and so phenotypes are really easy to look at and see. So they're going to be flower color, eye color, height. It's what you look like. Okay, so we can visibly see this on somebody. The second one that we're going to use is this term called genotype. Okay, and this is the actual genetic makeup. This is not something that you can see. You would actually have to be tested, or you'd have to have enough information about like the parents in order to figure out what the genotype is. Okay, and so this is, for example, what alleles do they have? So for example, if we're going to talk about the phenotype of the flowers, so we could say, well, let's look down here. Okay, so the phenotype of the flowers is that we have, some of them are purple and some of them are white. So these ones would have a purple phenotype. This flower would have a white phenotype. But we would then need to know what are the actual alleles, okay? And in order to do that, we would then have to go back to our example here and say, well, here is the genotype. So the genotype for this purple plant right here is capital P, capital P. These purple plants have a capital P, lowercase p. The big issue is that these look the same. They have the same phenotype, but they have different genotypes. And so it's often very difficult to know what your genotype is if you have the dominant phenotype. That's not the case when you have the recessive genotype or phenotype, because there is only one possible allele combination that would make a white flower. And so for this example, we would know exactly that this individual has a lowercase p, Okay, so it kind of depends on what you're looking at to figure out what is the genotype and the phenotype. Okay, these two terms are very, very important. You need to know the difference between genotype and phenotype. I think they're a little bit easier to understand because P, physical appearance, phenotype, G, genetic makeup, genotype. So they kind of go with what they are, okay? All right, so moving on from that. So Mendel's gonna do crosses with all those different traits that I showed you previously. So, you know, height and seed color and things like that. And he is going to kind of come up with these patterns of inheritance that he sees in all of them. Okay, so when he does these crosses of purebreds, he usually sees that one of the two traits disappears in the F1 generation, but then reappears in the F2 generation like we see here. Sorry, I keep on putting this. Okay, so doesn't matter what the trait is. If we looked at kind of tall versus dwarf, green versus yellow, pod color, any of these, in the F1 generation, the recessive trait is, you can't see it, but then it comes back in the F2 generation. Okay, so like this. So all of these ended up being purple, but then when you do this cross, you start to have this other ratio. So he did this experiment in all these different traits and found them all coming out and having the same exact ratio. Okay, and so the underlying mechanism of this explains Mendel's law of segregation. Okay, so pairs of alleles segregate during gamete formation. Okay, and then the fusion of gametes at fertilization creates allele pairs again. Okay, so we kind of already said that, but I just want to get this point across. Okay, so you kind of start off with individuals, and I'm just going to show them homologous chromosomes. So let's just say that, you know, they have these traits. Those are going to segregate during meiosis, where each sperm and egg is going to have just one version of them. You're also going to have the other. So let's just say this is mom, this is dad. Okay, they also are going to have, you know, homologous chromosomes that have the same trait because we're all the same species, so we have all the same genes. When they have egg and sperm produced, and then, you know, let's just say that this is an egg, this is a sperm. When these come together, they then are going to form a new pair of alleles, okay? And whatever that combination ends up being is going to determine what that individual looks like. Okay. So to kind of make that a little bit clearer, let me just write this down. Okay, so the relationship, between alleles and homologous chromosomes. Okay, so let me just get two different colored markers. Okay, so I'm going to draw a pair of homologous chromosomes right now. Okay, so hopefully these are, everyone remembers that these are the same, they're the same chromosome, but they could have potentially different versions of alleles. So what I'm going to do is I'm going to draw that they have some gene that we're interested in here, okay? And, you know, it could be eye color, for example. Okay, and so what we're going to call this location of a gene is we're going to call it a locus, okay? So a gene locus, I forgot to say this is homologous chromosomes. Okay, sorry. So then a gene locus is a specific location of a gene along a chromosome. Okay? And so then we're going to have alleles or alternative versions of a gene reside at the same locus. Okay, so like if we're talking about flower color again, purple versus white, it's at the same place on the chromosome. It's not like it's two different genes that are making one purple, one white. It's at the same place, and you just have one version that's going to create a gamete that, or a allele that would produce a purple flower, and one would be a white flower. Okay, and these are at the same locus on homologous chromosomes. Okay, because they are the same chromosome. They could potentially have different alleles, but not always. Homologous chromosomes. Okay. And just a reminder, okay, the two chromosomes may bear either identical alleles, in which case to be called homozygous, or non-identical or different, which is heterozygous. Okay, so what I wanted to kind of show how this looks, so I'm going to draw kind of my two homologous chromosomes again. I want to draw them bigger because I'm going to do a little more detail about this. Okay, because when we're talking about different traits and genes, there are, going to be multiple different genes on different chromosomes. Okay, so we're going to have kind of this red one. Actually, it doesn't really matter what color. I'll just draw them all the same. Okay, so the red just is showing where there is an allele that we're interested in. Okay, so these are 3 different genes that we're looking at. Okay, and what we can do is we can just say, what happens if we have different types of alleles on these ones. So for the first one, we're going to say that at this place on these homologous chromosomes, this one, and we'll just say this is mom and dad. We'll say that the allele for this one, you get a capital P, and on this one, they get a capital P, okay? And for the next trait, maybe it's a lowercase a, and they also have a lowercase a. Okay, so both of these have identical alleles at the loci. Let's say on this one, however, dad has a capital B and mom has a lowercase b. Okay, and so then what we can do is look at the genotype of this individual. So this is from dad and this is mom, but this is together you, or whatever individual we're talking about. So we could have big P, which is going to be homozygous. And because it's a capital letter, that means it's a dominant. So we're going to call that homozygous dominant. We could have little a, and little letters are always going to be recessive. So that means it's homozygous recessive. And then down here, we have a capital B and a lowercase b, which means that this is heterozygous. And we don't say recessive or dominant because it's going to have one of each. Okay? And so what we're looking at is kind of how the homologous chromosomes would hold the different alleles. They're going to have multiple different alleles. We could add another one up here and say, maybe this gene up here is going to have, capital T and lowercase t. Okay, and so then again, they would be, in that case, heterozygous for that specific trait. Okay, so these are the homologous chromosomes. So they are the same chromosome, but you got one from mom, one from dad. So what we're now going to start talking about is this idea of independent assortment, okay? And what I want you to start to look at is, you see how we're talking about, in this case, it could be 4 different genes that we're looking at, and they're all in the same chromosome, okay? And so we want to know, do these different genes affect which ones you'd get? So for example, we have a capital P here, a lowercase a here, and a lowercase b here. When this chromosome gets passed on, do these have to stay together, okay? Or is it possible that, we've talked about crossing over, that you could actually get these different combinations of the alleles, okay? And so this whole idea is going to be called independent assortment, okay? So Mendel's law of independent assortment, okay? And so the first big thing that's going to happen is that we're now going to start talking about 2 different characters. So we're not going to just talk about flower color. We're going to start talking about, for example, what happens if we do seed color and shape, okay? We have to start talking about 2 traits together, okay? So before I kind of get into that, I just want to talk about the previous examples that we've done. So if you do a cross between 2 individuals that are heterozygous for 1 character, it's called a hybrid cross, okay? Because that means you're looking at just 1 trait. What we're going to talk about now is something called a dihybrid cross, okay? So we're going to be looking at 2 different traits together, okay? So it's going to be a cross between 2 organisms that are heterozygous for 2, this keyword, characters. So basically what Mendel was starting to look at is that, are characters linked? Okay, so for example, if we have a purple plant that is tall, and those are pure breeding, and we cross it with a white and short plant, what are the possible offspring? Are we only ever going to see purple and tall together? Or is it possible that you could see white and tall or purple and short? Okay, so could you have purple and tall plants and just white and short? So you basically have parental characteristics with these 2 traits. Or is it possible that you could have a purple short plant and a white tall plant come out? And so basically taking parental characteristics and seeing what are the possible outcomes when we look at 2 different traits at once. And so Mendel had 2 hypotheses that potentially could have happened, okay? And so he then did the experiment, but his 2 hypotheses are important for us to kind of talk about. Okay, so his first hypothesis was that if genes for 2 characters were linked, so if those 2 characters were linked, so if purple and tall were linked together and white and short were linked together, then the F1 hybrids would only produce the same 2 kinds of gametes that they received from their parents. So basically, purple and tall would stick together, and white and short would stick together, okay? Which would then mean in the F2 generation, you would have kind of this 3 to 1 ratio that we saw in the monohybrid cross. Or, in spoilers, this is not what happens. This next one is, so if the two characters sorted, which basically means separated, independently, then the F1 generation would produce 4 gametes and genotypes in equal quantities. And then the F2 generation would have 9 genotypes producing 4 phenotypes in a ratio of 9 to 3 to 3 to 1. Okay? And so that would mean basically 9 different genotypes, so allele combinations, and 4 different phenotypes of what they actually look like. Okay? So this might sound like a bunch of nonsense. The best way to actually kind of do this is to actually do the cross, okay, and show you what this would look like. Okay? And so I'm just gonna tell you ahead of time that there is independent assortment. So I'm going to show you what that looks like. Okay. So here is our dihybrid cross. So we're going to talk about yellow round seeds, and we're going to have green wrinkled seeds. Okay. So we have pure breeding yellow round seeds and pure breeding green wrinkled seeds. Okay, before we go too much further, I want to define what letters we're going to use. And so we're going to use the letter R for round, okay, and the round is dominant, okay, which means that wrinkled is recessive, and yellow is going to be dominant. Okay, and so our letters that we're going to use is, so we're going to use capital R for dominant round, and we could have our different kind of genotypes. Okay, so these two would both be round, and this would be wrinkled. Okay, and for yellow, we're going to use Y. Okay, so we have capital Y, or capital Y, little y. These two are going to be yellow, okay, because remember, homozygous dominant or the recessive, the dominant trait will show up in the heterozygote form. And we'll have little y, which is going to be green. So we have yellow round, and these are pure breeders. So you can never have heterozygotes when you have pure breeders. So this is what this individual is going to look like. So they're going to be big R, big Y, because they are round and yellow. For this group, they're going to be wrinkled, which is little r, and they're going to be green, which is going to be little y. So this is what the cross is going to look like. So again, we now have more letters because we're talking about 2 different traits. Now, when we're doing this first part, we have to figure out what are the different combinations of the gametes that we can make. And so for these, because there's only 2 different letters, all of our gametes are going to have a big Y and a big R, And all of these gametes over here are going to have a little y and a little r. Okay? So when you do the cross, basically all of the individuals are going to come out looking like this. And I can actually do this one out for you if you need me to. I'll probably do this for lab, show this cross, but basically they all turn out to have this kind of genotype, okay? And so all of the offspring are going to be this. And so big R, little r is going to be round. So this would be a round, and big Y, little y is going to be yellow, yellow. Okay, so for the F1 generation, all of them are round and yellow. So just this one parental type. What is interesting is when we start to do a cross between these groups. Okay, so we have a big R, big Y, little y, and cross it with a big R, big Y, little y. Okay, and so when we're doing this, we can then, what are the actual gametes that we can make? Okay, and we have to have one R and one Y for our gametes. So we can have a big R and a big Y. We also can have a big R and a little y. So I'm going to do that. We can have a little r and a big Y. And we can have a little r and a little y. Okay. Same thing over here because these are identical. So big R, big Y, big R, little y, little r, big Y, and then little r, little y. Okay. And I like to just draw these in circles because it makes me think, oh, these are sperm or egg. Okay, so these are going to be our different gene combination, alleles in the actual sperm and egg. Okay, so I'm going to start on a new page because I need a lot more space to do this. Because this is going to be, I'm going to start making a Punnett square. So I want to show you what this will look like. Let me just rewrite this. So this is the cross that we're doing. This is the dihybrid cross because there's two different hybrid groups going on. Okay, and I'm going to draw a large square. Okay, and so this is going to have 16 boxes. And so what we just looked at, we found out all of the different allele combinations that we can make for our gametes. And so for both of them, these were our different types that we can make. Okay, and so when you have the different gametes that a specific kind of genotype can make, you then put them on the different sides. Okay, so we're going to have like this. And because of the same, I like to write them in the same pattern because then it makes it easier to look at later on. Okay, and so these are the gametes. And so what we could say is like, these are all the sperm and these are all the eggs. And so when they come together, what is the genotype that they're going to create? Okay, so I'm going to go through and I'm going to look at, okay, what are my R's? And then I'm going to look at my Y's. Okay, and then I'm going to say big R, big Y, little y. Big R, little r, big Y, little y, or big Y. And I'm just going to go through and do this. Oops, that is not correct. Oops, that was supposed to be a capital Y. I need you guys here with me so you can tell me when I'm doing things wrong, which is all the time. All right, hopefully that is all correct. Okay, so luckily you won't have, I will never make you do one of these on an actual exam. But this is what we would call a dihybrid cross. So it is more complicated. So the next step is to then go through the actual cross and see, okay, how many different types of genotypes do I have, and what are the different phenotypes that would have? Now, what I want you to start to look at is that there are some patterns. So you have more than one of some of these. So I want you to notice that all of these are the same. Okay, so those are all going to be the kind of big R, big Y, little y. Okay, and then, so we can kind of write that out. And then you could look for other patterns. So these two are the same, these two are the same, this one and this one are the same, this one and this one are the same. And then we have a couple that are not going to have any matches. Okay, and that just kind of happens because of this pattern that we have. But basically what would happen if you counted all of these out, you would then end up with a 9 round yellow. Okay, and remember, you just go to each one and say, okay, we have round is dominant to wrinkled. So anytime you have a capital R, you know it's going to be round, and yellow is dominant to green. Okay, so we would then have 3 round green. Okay, so that is now a new combination that we did not see in the parents. We would have a wrinkled yellow. That also has not been seen in the parents. And we have a wrinkled green. Okay, so these two right here, the 9 and the 1, round yellow and wrinkled green, those are the parental types that we started with. Okay, because we said yellow round, wrinkled green, okay? But these two right here are the ones that are showing us that these are not linked genes. So not linked, because you can get all different types of combinations. So let's write that out. Okay, so Mendel's dihybrid cross supported the hypothesis that each pair of alleles segregates independently of the other pairs during gamete formation. So basically what it's saying is, in that specific case, color of seed does not affect kind of shape of seed. Okay, so they are not linked together. Okay, so basically, big important statement, the inheritance of one character has no effect on the inheritance of another. Okay, and so this is basically the law of independent assortment. All right, so Mendel now has kind of these two major laws. Oh, and before I move on, I just kind of want to show you what this actually would look like. So if we were, this is from, you're going to have all of this I gave you in another PowerPoint. But this is basically just showing you what the different things would look like. Okay, and so you can see kind of the different generations. Okay, so moving on from peas, one thing, I know most of you are not going to be breeding peas at any point, but one thing that can happen, and if you've ever had anything to do with dogs or dog breeding, you might run into the situation where you don't really know the parentage of a certain dog or cat, and you'd be interested to kind of know what their different genotypes are. And there's actually a way of doing that without having to do a genetic test. And so what you can do is something called a test cross. Okay. And most people just wouldn't do this, but this is just something that you could do if you wanted to. So a test cross is kind of a mating, between an individual of dominant phenotype but unknown genotype and a homozygous recessive individual. Okay, and so a good example of this has to do with Labradors. So in Labradors, there is kind of, you can have lots of different coat types, but for example, this one type group, you can have either black labs or chocolate labs. And if you were trying to maybe breed, you wanted black dogs instead of brown dogs, but you didn't know if one of the dogs, what its actual genotype was. You knew it was a black dog because you can see physically that it's black, but you have no idea if it's gonna, if it has a big B or a big B little b. Okay, and if you want to breed one specific color, it's important for you to know what their actual kind of genotype is. Okay, because if this is a heterozygote, you'd probably not want to use this individual to create black dogs because you're going to end up with having brown dogs. And so what you can do with a test cross is you can take kind of your unknown, so it has the dominant phenotype, but you don't know what its genotype is, and cross it with a homozygous recessive. And so in this case, brown is recessive to black. So if you mated these two, there's two possible things that could happen. The first thing that would happen is that if all of the puppies were black, then you would know that this individual was actually a capital P, capital B, so homozygous dominant alleles. If you, however, ended up with half and half, black and chocolate labs, then you would know that this individual was actually a heterozygote and had a big B, little b. Okay, and so it's able to actually, you can use this test cross to figure out what the actual genotype of an individual is. Okay, now one thing I wanted to mention very briefly, we're not going to talk about this very much, is this idea of the rules of probability. So Mendel was an excellent statistician and mathematician. And one of the reasons why he was so kind of good at genetics, probability, well, let me just put this out before I can't speak in right at the same time. So one reason why he was so good at genetics is because genetics is all about probability, okay? So basically the patterns of inheritance have to do with probability, okay? And like I mentioned before, when you're thinking about having a child, it's a 50-50 chance if it's gonna be male or female. And if you have one female one time and another, your next baby, those two things do not influence each other at all. So each time is gonna have kind of, it's equal chance of getting a boy or a girl. Okay, and so genetic crosses are going to obey the law of probability. So genetic crosses obey, rules of probability. Okay, and so one of the rules that's really important in kind of genetics is the rule of multiplication. And what this does is it states that the probability of a compound event is the product of the separate probabilities of the independent events. Okay, so what this compound event thing means, so for example, if I give you a quarter and I say flip the quarter once, what is the probability of you getting a heads or a tail? Okay, and then I say, well, what's the probability of you flipping 2 heads in a row? Okay, so each event independently will have a 50-50 chance of heads or tails, and then in order to see like what's the chance of you getting a heads and a heads, you would then have to multiply, well, it's 50-50 chance for first heads and a 50-50 chance for the second heads, and so therefore you multiply those together and it's going to be a 25% chance that you get 2 heads in a row. So here is just kind of an example of what this would look like with coins and also kind of phenotypes, is that you can kind of look at the different gametes that you have from the individuals, and in this case they're both heterozygotes for big B, little b. And so you end up kind of seeing, well, what is the probability that you'd end up with, you know, 2 big B's or 2 little b's or a big B and a little b? Okay, and you can have these different probabilities. So there's a 50-50 chance that from the mother you're going to get a big B or a little b. There's also a 50-50 chance you're going to get a big B or little b from the father. And so 50-50 chance, so big B would be 1/2 times 1/2, which is equal 1/4. Okay, so I'm kind of rushing through this because we'll talk about it a little bit in lab, but one of the things that comes up in lab is this idea of what's the probability, the probability of having 3 boys in a row. Okay, and so I want to talk about this one because this one actually you'll see again. But if each reproductive event, okay, you have 1/2 chance it's going to be male for the first event. Okay, so that's child number 1. Child number 2, 50-50 chance, male or female. That's number 2. And the third child, 50-50 chance, it's going to be male or female. Okay, and so it's always going to be a 50% chance that it's going to be a boy. So to have 4 boys in a row, you take 1/2 times 1/2 times 1/2, which would be 1/8 of a chance. Okay, and so that would be how many times, or the probability of having 4 boys in a row if you're having babies. Okay, it would be the same thing if you were to say, well, what is the probability of having three girls in a row? Same thing. And then it could say, what is the probability of having 4 boys in a row? All you would have to do is add an additional 1/2 and do that math out. Okay, so 1/16. So we're gonna, again, very briefly just talk about that, but just realize that everything in genetics is gonna have to do with probability.